cell cycle and cell division. So here any living organism starts its life with a single cell structure, but if it is a unicellular organism, the number of cells remain same, but if it is a multicellular organism, the cell which have been started its life, that will start repeatedly dividing to form a multicellular organism which contains a millions of billions of cells. So here as the cell is repeatedly dividing to form a multicellular organism, so that process of division is considered, which will be taking place in a cyclic manner. We call the changes from one division to other division as a cell cycle. So in this process many such changes will be taking place. Before that, the concept of cell division was given by Rudolf Virchow, scientist. So he had given a concept called as cell lineage theory or cell doctrine. According to him, omnis cellula e cellula, that is new cells arise from pre-existing cells by a process called as division. So that is the cell is undergoing division. That concept was given by Rudolf Virchow, scientist, for the first time. So likewise, from when a cell is dividing, new cells are formed. From the formation of new cells to further the cell, once again after its growth and development, further it will divide. So the changes from the one division to the other division is called as a cell cycle. So as I said that every living organism will start its life from a unicellular structure called as zygote. So this is an unicellular structure and diploid in its nature. This zygote will undergo division. That is what we call it as cell division. And not only cell division, whatever the cells which have been formed, they have to increase their size and acquire the normal size of as like that of a parent cell. So that is called as cell enlargement or elongation. And not only the cell elongation, cell differentiation will also take place. That is some of the group of cells will be converted into some specific part of a organism, like the cumulative effect of all these three. The cumulative effect of all these three lead to the, that is nothing but the growth, lead to the formation of an organism. So in this concept, cell division is the basic thing which is taking place during the formation of a zygote to a complete organism. So here all these changes which are taking place, like what are the changes that are taking place during the cell cycle means, so the cell is dividing to form 2 daughter cells, or depending upon the condition 4 daughter cells may be formed, and new cell organelles are formed. The duplication of DNA molecule, the increase in the number of chromosomes such that the number will be distributed equally between the daughter cells. Such type of changes which are taking place in the due course is called as a cell cycle. Okay. Various processes or various changes which are taking place during the cell cycle are controlled by protein cyclins. That is cycling dependent protein kinases. We call them as CDKs. So this and apart from this for CDKs, so here the energy is also being required, which is provided in the form of ATP molecule, that is adenosine triphosphate molecule. And here there are some checkpoints also been there. There are 2 checkpoints are there. So during the process where checkpoints are there between the G1 to S phase and G2 to M phase. So this is called as CG1 and CM. The checkpoint between G1 to S is called as CG1 or G1 cycling or cycling G1, whereas the checkpoint between G2 and M is called as mitotic cycling or CM. And this entire process of checkpoints are also being mediated by the energies. So here the process from G1 to S phase, when the cell is in the G1 phase, whether the cell have to enter into S phase or not, so that will be decided by the checkpoints, whereas when the cell enters into G2 phase, it have to enter into the mitotic phase will be decided by the checkpoints, that is CM or mitotic cycling. So here how they are operated, let us see in the form of an diagrammatic representation, whether how this cell cycle is taking place and where are the checkpoints are operated for the entire process of cell cycle. We know that in the cell cycle, interphase and the mitotic phase are present or the division phase are present. So interphase is divided into 3 stages. Those are called as G1, G1 phase and S phase and third one is called as G2 phase and the mitotic phase. So here the cyclings or checkpoints are present between G1 S and the G2 and M phase. So let's observe the steps that are taking place here. The cycling dependent CDKs, cycling dependent protein kinases. So these are the one which are in inactive stage initially. So it contains the inhibitory site and also the activation site. This is the inhibitory site and this is a activation site. So initially it will be in the inactive stage, but it will become active by taking up by utilizing the energy present in ATP molecule, where it is converted into ADP molecule. The checkpoint CG1 will get operated here, which enter into the scene, and where so it will be converted, where it will the checkpoint CG1 will come and attach to the activation site, so that the inactive CDKE now it becomes active. When it become active, then only the G1 phase enters into the cell cycle. From G1 phase to S phase will be proceeded. Otherwise, if it is not activated by the G1 cyclin and ATP, the CDKs remain inactive and the cell cycle will be stopped from G1 to S phase. So here we can show the diagram in the form of like this, where inactive stage and the active phases, where the phosphate group get attached to the phosphate. From where does we get the phosphate means the phosphate from the ATP, that is adenosine triphosphate, is losing one inorganic phosphate to form ADP. That inorganic phosphate will come and attach to the activation site of the CDKs. So with the help of that, the CG1 will attach to the CDK, so that now it becomes activated, then only this will become active. Such the cell cycle will be moving from G1 phase to the S phase. And in the process, what happens? So this inorganic phosphate will be lost, from this one, and whatever the CG1 that is present, that will be broken into small fragments. And now once again this CDK is available for the next step. Now the checkpoint 2, from it here it will enter into the G2 phase, where once again the ATP will be providing the energy. So here 2 ATPs are taking part, because it requires 2 phosphate molecules. Mitotic spindle. So here whether the cell cycle have to move from G2 to M phase, that will depends upon the mitotic cycling. So the mitotic cycling will also comes and operated in the process. Mitotic cycling. Here 2 phosphate molecules which are there, they will get attached to both to the inhibitory site and also to the activation site. Once they attach to the inhibitory site and the activation site, 1 phosphate on the activation site and 1 phosphate on the inhibitory site. So here this is a CM. Now this CDK is in the inactive stage. Why it remains in the inactive stage means because of one of the phosphate is attached to the inhibitory site, but once it have been lost, once the one of the phosphate had been lost from here, now it become active, because now the phosphate is present only on the activation site. So here further the phosphate molecule, one more phosphate which is that is also been lost, and the mitotic cycline will also be broken. So this is the way how these 2 checkpoints. So this is the first checkpoint and this is a second checkpoint. The first checkpoint is helping in the checkpoint which will control the whether the cell cycle will move from G1 to S phase, and the second checkpoint will check the cell cycle to move from G2 to the M phase. So these are the changes which are taking place, which have been controlled by the choline dependent protein kinases along with the ATP and the G1 cycline, which is acting as a first checkpoint, and the C1 which is acting as a mitotic cycline, which is acting as second checkpoint in the entire process of cell cycle in a living organism.